Going to transition over now. So our next speaker, our next contributing speaker today is my colleague, Dr. Oskar Stauffer from the Max Planck Institute for Medical Research in Heidelberg. Dr. Stauffer will take a moment to introduce himself further, and then he will present his work on the bottom-up assembly of functional, fully synthetic extracellular vesicles for 15 minutes, followed by 5 minutes for questions. I'll let you take it over now, Oskar.
Thank you very much, Jacqueline. So I hope this is working. Yeah, so my name's Oskar Stauffer. I'm a postdoc currently in the Department for Cellular Biophysics at the Max Planck Institute for Medical Research in Heidelberg. I am a biotechnologist by training, and my recent work has mostly focused on applying concepts from bottom-up synthetic biology and transitioning them into more biomedically relevant questions. And this is also what I will be talking about today. So as a general introduction into the field, I think all of us who work and study living systems, we can appreciate that nature has the power to transform a molecular complexity and molecular diversity into very elegant, effective living systems. So what I mean by this is, if we look at structures like the ribosome, they're obviously very complex in their dynamics. Even though they're not composed of many different components, we cannot yet fully understand which part is responsible for which kind of a function. If we now imagine these molecular machines also in living cells, we can, I think, appreciate, especially in the form of collective cell migration, that cells display a spatio-temporal diversity. So they coordinate their actions between each other. And when we come to this stage of complexity and of molecular diversity, it's even harder for us to follow how nature works and how cells actually live. So the general approach in the field of bottom-up synthetic biology is to perform in vitro reconstitutions of biomolecules in order to understand how the fundamentals of life actually work. So what we try to do is to reconstitute low complexity systems and analyze their behavior in a well-controlled microenvironment, like you can see in this example of microfluidic droplets that are forming tubulin esters, like we see them during cell division. So the process I'm mostly interested in is, again, a very complex process. It's a regenerative process, especially wound healing, or particularly wound healing. I think if we look at such a time lapse of wound healing, we can all appreciate that the regeneration of our skin is very complex and very dynamic. Of course, years of research has now told us that many cell types within such a wound are responsible for the regeneration. We know that there are epithelial cells that will migrate into the wound site. We know that there are fibroblasts and other connective tissue cells that will be able to regenerate all the connective tissue. We know that there are blood vessels forming, and we know that there's this immune reaction on site that will prevent invaders like bacteria to enter the wound. So we know that wound healing is a very complex process that is based on the coordinated interplay between distinct cell types. Now the question arises, how do these cell types coordinate themselves, and how do they coordinate their multicellular actions? One of the key players in this process has actually turned out to be extracellular vesicles, more specific exosomes. And for those of you who are not familiar with extracellular vesicles, I just want to give a brief slide on why they are important and why we should be studying them. So extracellular vesicles are lipid-based vesicles released by cells. They can either be of intracellular origin, like exosomes, or they can bud off from the plasma membrane of cells in the form of microvesicles. What is most important about them is that they carry a biomolecular cargo that can be proteins or any kind of a genetic information like messenger RNAs and microRNAs. And these extracellular vesicles, when released by a cell, are able to travel to a distinct cell and regulate the functions of this cell. We know from, let's say, transcriptomic and proteomic approaches that these extracellular vesicles are, again, very complex. They contain different kind of proteins. Up today, we know more than 40,000 different proteins that can be found in these extracellular vesicles. We know that they contain many different kind of RNA molecules like messenger RNAs and microRNAs. And what is most important, we know that they have a very diverse functional spectrum. We know that we can apply these extracellular vesicles to regulate antigen presentation. We know that they have an important function in cell migration and bone resorption and in many, many other biological processes that are not only interesting to us as scientists, but also very interesting for therapeutic approaches in the biomedical sciences. So this means that new insights into the biology of extracellular vesicles will not only improve our understanding of intracellular signaling, but will also enable us to develop new biomedical applications. Mostly extracellular vesicles have been in the focus of diagnostic approaches as they mirror the physiological state of the cells of origin, and they can be easily recovered from bloods and other biofluids. But there are also several approaches that focus on the application of extracellular vesicles as kind of endogenous regulators of physiological states. This means extracellular vesicles are purified from a patient or a donor and then re-administered into a patient in order to perform a therapeutic function. However, both approaches, and especially the therapeutic approach, is still limited by several factors. For instance, there are fluctuating production conditions. These productions are also very time-consuming and error-prone, which is the main reason why there's no EV therapeutic that is currently approved by regulation authorities, neither in the US nor in Europe. And of course, the complexity and diversity of these extracellular vesicles are still impeding our quantitative understanding of exosomes, for instance, and also impeding us as engineers and bioscientists in programming the functions of these exosomes, either by bottom-up approaches or by top-down design of their functions. So the main aim of my work that I would like to present you today was the bottom-up assembly of fully synthetic extracellular vesicles that circumvent all of the previously mentioned limitations. That means I want to build extracellular vesicles and assess their therapeutic functionality for a more systematic analysis of their functional signaling mechanisms. This means instead of going the conventional way that's depicted on the left side of this image, where extracellular vesicles are isolated from a biosource and we obtain, let's say, vesicles that are poorly defined on a molecular level, I would like to start from the individual molecular building blocks, that is lipids, RNAs, proteins, and so on, perform a very controlled in vitro assembly in order to obtain vesicles that display a comparable or equivalent therapeutic response in cells. So most specifically, this means the specific goals are the bottom-up assembly of fully synthetic wound-healing extracellular vesicles and the assessment as well as engineering of their therapeutic functionality. So just a brief slide on how we perform this. Of course, if we want to construct such, let's say, still complex supramolecular compartments like extracellular vesicles, we need very sophisticated manufacturing approaches that allow for a controlled in vitro assembly. For this, we mostly use microfluidic technologies as well as other emulsification techniques in order to produce water and oil emulsions that we can use as kind of scaffolds to build up extracellular vesicles from their lipid and protein and RNA components. The basic process is shown in this figure, but I don't want you to go through all of the details at this point. The important thing is we can have a high and very precise control over the structure as well as the composition of the synthetic extracellular vesicles. To give you an idea how synthetic extracellular vesicles look like in our hands, this is a confocal microscopy image of some of these vesicles. You can see they're surrounded by a lipid membrane, just as their natural counterparts. They have specific proteins on their surface, and they contain, in this case, microRNAs, so regulatory elements in their lumen. What I mean when I say that these structures are more defined on a molecular level and that they're less complex is something I would like to show you by their simple size analysis. If we look at extracellular vesicles that are isolated from living cells, we usually see that there's all kind of different populations of extracellular vesicles that can be simply discriminated by their size. Of course, this is a hurdle if you want to understand the contributions of every single population for a specific therapeutic effect. And here's a size analysis of our fully synthetic extracellular vesicles. You can already see they're way more defined in terms of their size. And this also applies in their molecular structure. So the question, of course, is, do these fully synthetic extracellular vesicles still interact with living cells in a comparable way to their natural blueprints? And this we analyzed by several techniques. I'm just showing you here some fluorescence microscopy images, as well as some electron microscopy on the lower part of this figure. And you can see that the fully synthetic extracellular vesicles that are shown in yellow, they attach to the target cells and they're uptaken into the interior of this, into the interior of the target cells and release their regulatory elements, so in this case, microRNAs, into the cytosol of the target cells. So of course, if we want to prove that fully synthetic extracellular vesicles are able to induce therapeutic functions in target cells, we need to come up with specific molecular blueprint that allows us to reconstruct exosomes, in this case, in a fully synthetic way. And by this, we turn to a publication from scientists in Berlin that described human fibrocyte-derived exosomes that were accelerating wound healing in mice. The authors of this
Oscar,
study—
you have 5 minutes. Sorry to interrupt you. 5 minutes.
The authors of this study pinpointed 3 specific effects, phenotypic effects that they can measure in the target cells, which is an increase in proliferation as well as increased migration and collagen deposition by the cells. And they describe some of the most important components of these extracellular vesicles, which are 6 different kinds of microRNAs and 3 types of transmembrane proteins, tetraspanine proteins. So we went on to simply reconstruct by this molecular blueprint the extracellular vesicles and test their therapeutic functionality in living cells. I'm just showing you here how the fully synthetic extracellular vesicles increase the collective cell migration of cells. These were measured by in vitro assays of wound healing behavior of keratinocyte monolayers. And then we basically sequentially increase the complexity of the extracellular vesicles that we applied, basically by testing one tetraspanine molecule after another, or basically all of them in a combined way, and then successively adding microRNAs one after the other into the lumen of the extracellular vesicles and measuring the effect and the contribution of every single molecular component to the overall effect. So by this, we concluded that the extracellular vesicles that we designed increase the artificial wound, or the healing of the artificial wound sites. We obtained comparable results of this for the collagen deposition as well, for the increase in proliferation. I'm just not showing them today for the sake of time. Most importantly, we also could prove that the fully synthetic extracellular vesicles improve or induce angiogenesis in 3-dimensional human skin models. So what you can see on the upper part of this image are synthetic vasculatures in human 3-dimensional skin models. And we tested also the wound healing of these 3-dimensional skin models with human donor skin. And we could see that basically when we treat wounded skins with fully synthetic extracellular vesicles, we can increase the wound healing strength and speed of these keratinocyte layers in the wound by the treatment of our extracellular vesicles, meaning that the fully synthetic extracellular vesicles that we produce display a comparable therapeutic functionality to their natural blueprints. Now, starting from here, this was all very much focused on, let's say, biomedical therapies and their industrial application, but we can also contribute better to an increased understanding of the molecular signaling pathways of these exosomes. For instance, by applying then a combination with transcriptomic sequencing, RNA sequencing of the target cells. So we basically analyzed, based on RNA sequencing, what kind of specific pathways, biochemical pathways, are activated by treatment of the fully synthetic extracellular vesicles. And in contrast to the natural fully synthetic extracellular vesicles, we were now able to assign every specific activation of specific biochemical pathways that promote healing of the wound site to specific molecular components within the extracellular vesicles. This is something that could not be achieved by classical top-down, let's say, knock-in, knock-out approaches very easily. Just as one example, we could see that a combination of the tetraspanin 1 and 3 is responsible for the activation of all kind of immunology-related processes in this case. So as a conclusion, what I showed you today is the design and the assembly of programmable synthetic extracellular vesicles that very accurately mimic the functional therapeutic spectrum of natural extracellular vesicles. I showed you how to identify the key microRNA and protein composition of these extracellular vesicles and how to assign them to specific biochemical pathways that they trigger. And at the end, I would like to highlight that the fully synthetic extracellular vesicles that we assembled from the bottom up overcome the systematic limitations of natural extracellular vesicles by breaking down the complexity and the diversity into a more, let's say, comprehensive level for us scientists. In the future, I think this has the potential to bring forward a completely new class of therapeutics that are based on merging synthetic, very much lifelike structural assemblies and living systems. And also it has the potential to create synthetic communicative environments for living cells and thereby dynamically regulate their phenotypic adaptations and alterations within in vitro or in vivo systems. At the end, I would like to thank all the people that were involved in this work. This is mostly my supervisors, Joachim Spatz and Ilja Platzmann, as well as all the people in our very own institute at the Max Planck Institute for Medical Research and all the partners all around the world. Let's say the funding agencies, especially HPICS and the Max Planck School of Medical Life as my graduate schools, and the Max Planck Bristol Center for Minimal Biology that we are having currently together with the University of Bristol. And at the end, very much thanking you for attending today, and I'm open for questions.
Great, thank you very much, Oscar. So we do have a few questions from the audience. The first is from Frank Lee. He asks, in what ways are the mRNA COVID-19 vaccines similar and different from extracellular vesicles found in nature?
Well, it depends on how you define difference and equality in this case, of course. Yeah. So the current vaccine strategies are also based on liposome technologies. They harbor, in this case, messenger RNA mimetics in their lumen, and they also deliver a therapeutic cargo to the target cells, in this case, most likely dendritic cells. Of course, compared to natural extracellular vesicles, they have a way more reduced complexity because they're basically only composed of some synthetic lipids and the synthetic RNA molecules in their lumen. At the end, their functional spectrum, I think, is not as large as those of natural extracellular vesicles, as well as those of the fully synthetic extracellular vesicles I presented you today.
Great. Okay, next question from Cynthia Colejo. She thanks you for the great talk. She would like to know if or how the extracellular vesicles are directed to their target.
Yes, so in this specific case, we don't know yet. Yeah, we have, as I mentioned, 3 tetraspanin molecules on the surface of the extracellular vesicles. This might already provide some targeting functionality. Although the tetraspanin molecules are not very exclusive for this set of exosomes that are presented today, still they might influence or at least define somewhat of a spectrum of possible interacting cells to which they're then targeted.
Okay. David Gonzalez asks, do you notice any difference in the efficiency of cellular uptake of the EVs depending on their size relative to the cell?
Yes, that's a short answer, yes. Of course, how much extracellular vesicles, either natural or synthetic ones, are uptaken by cells is, first of all, very much determined by the biophysical features and the biochemical activity of a cell. For instance, epithelial cells in most cases display reduced uptake compared to fibroblasts or macrophages. That's partly, or can be partly attributed to their membrane topology as well as membrane turnover rates. And of course, it's also somewhat defined by the size of the vesicles that we apply. Larger vesicles usually experience lower uptake rates.
Okay. And Dana asks, what are the challenges in making such vesicles?
Well, on the industrial level, of course, it's the major challenge is to produce large amounts in short time, which is something that this technology can achieve. But it's also, of course, when it comes to the point of getting approval from drug agencies, you want to produce them in a very defined, narrowly defined composition and in a very high, pure, and reproducible composition. This is for the industrial, let's say, application spectrum. If you want to do this to get to know them a little bit better and to understand how natural extracellular vesicles work, I guess the major challenge is in engineering these vesicles on a quantitative level, I would say. So, of course, you want to fine-tune the ratio of, let's say, tetraspinin 1 molecule to messenger or microRNA 4 molecule in a very quantitative and reproducible manner. Because this opens up far more possibilities than simply checking basically on a qualitative level which kind of molecules are allowing for which kind of a function in a cell.
Great. All right, last question from Pasquale Stano. He says, congratulations, it is a beautiful research. Is it important or not to reduce the size of fully synthetic extracellular vesicles for some specific functions? And also, what about hybrid extracellular vesicles by mixing natural and synthetic systems? And he thanks you.
Maybe let me start with the last question. Of course,
Sure.
in general, sorry?
Go ahead.
Okay, sorry. Of course, creating hybrid systems can open up new possibilities, basically by taking the advantage of what natural systems already provide to us. But in some instances, of course, this might, let's say, trouble a little bit our requirements when it comes to defineness as well as to reproducibility of the structures that we want to build up. So it's, let's say, something that you would need to judge on an individual basis depending on what kind of an application you're looking for. And the first question, could you repeat the first question? I already forgot, sorry.
Sure, no problem. The first question was, is it important or not to reduce the size of fully synthetic extracellular vesicles for some specific functions?
Well, the size of the extracellular vesicles, we already mentioned that they determine uptake rates, for instance. They can also influence how specifically the proteins on their surface signal with cells. But of course, when you think about the in vivo situation, the size will not only limit the uptake of the vesicles, but also somewhat be important for the physiological and anatomical barriers that we face under in vivo conditions. So smaller extracellular vesicles usually display longer circulation times when injected into the bloodstream. So that might be an important factor. Of course, they also diffuse faster through connective tissue. This is something that might be important for a particular application. Well, and so on.
Sure. Okay, great. Thank you very much for your answers to the questions.
You.
Okay, so with that, I will go ahead and make the announcement for next week's speakers on April 5th. at 10 a.m. Mountain Daylight Time and 6 p.m. Central European Time. We will have Doctors Eberhard Bodenschatz from the Max Planck Institute for Dynamics and Self-Organization, as well as Dr. Lado Otrin from the Max Planck Institute for Dynamics of Complex Technical Systems. So we thank our speakers for the wonderful presentations today, and hope to see you all next week. All right, thank you.
Bye-bye.
Bye.